So I'm going to talk a bit about Bionodes, but before I'm going to give a bit of context. So I'm a PhD student at Queen Mary University, Worm Lab, and I'm also a Mozilla Science Lab fellow. So we're trying to promote open science and open source in academia. And the problem is this. So this is the volume of genomic data that's being generated. And if our predictions are correct, it's going to surpass everything. In this paper, you can check the link. So the slides are all online, and you can look at the paper in detail if you want. But they're predicting it's going to surpass astronomy, Twitter, YouTube. We're going to be generating zettabytes, exabytes of data a year. And this is not fiction, because for my PhD thesis, my raw data compressed. So the things that I took at the start was 7 terabytes compressed. And I'm currently using 40 terabytes on the cluster at the university. And the machines I typically need to run this stuff have 500 gigs of RAM. And we're going to get now new machines with 2 terabytes of RAM. So how did this happen? Well, there's 2 reasons. One is the cost of sequencing that's decreasing. And it's decreasing a lot due to the new improvements in sequencing technologies. And with each generation, it keeps decreasing. And we soon will have sequencers. We already have sequencers that size. You can just plug in your laptop. And they're working on sequencers you can plug on your phone. So sequencing is going to be accessible to anyone. And this means we're going to be flooded with genomic data. And the other problem is usually bioinformatics software sucks. And the reason for that is that the incentive in science is to publish papers, not to publish beautiful code or good code, or to make your tool reusable a lot. So science is kind of broken right now, but at the same time, there's a lot of people trying to fix it, and that's what we call open science, and we're trying to promote open source and best practice in science. So what Bionode is trying to do is build tools that try to suck less. And for that, we're trying to make modular and universal bioinformatics. So we're trying to create tools that try to do one thing well, provide highly reusable code and tools so that we don't keep reinventing the same functions in every project, and be able to scale with big genomic data, and also try to run our code everywhere, because it's really hard. We need to use to rerun our experiment across several machines, and it's really hard to get all the things you need to run on those machines. So we're using things like Docker a lot and virtual machines. But the cool thing is that if we use node.js, we can have highly modular code. There's a lot of modules on NPM. The community is very open and everything is on GitHub, so it's really easy to contribute and change things. And Node.js provides a native implementation of streams, which we want to use to scale for big genomic data. And also, since it's JavaScript, you can run it on the browser and on the command line. Not that you would want to run 500 gigs of RAM analysis on your browser, but there are small things you can do with web apps, or you can look at a sequence and do a small analysis. And right now, Bionode is an open source community, so some people we basically rely on contributions. We have a few people that are more or less active on the project, and we also we are being used in a bunch of other web apps and tools and projects on some universities, but also there's a startup in Cambridge using Bionode. Some of them are here. You should talk to them later. And there's also other open source projects using Bionode. We currently have around 13 tools, but a lot of them are still kind of work in progress. Also, try to get students for Google Summer of Code to work on Bionode for 3 months. So we got one last year. We're going to get another one this year. So every year we try to apply, and until now we've been successful. But the message is we need more contributions. So if you want to have a look at this, if you're interested, just check our project board and pick an issue or come talk to us. That was the introduction to the project. So how do we use streams in this project? Well, I'm going to give an example of one of the modules. The Bionode NCBI module is to access a database from the National Center of Biotechnology and Information. And what we do is we require this module, and then usually in JavaScript you use a callback pattern. So that means you get all the data, and then once you get all the data, you do something with it. But if you use an event pattern, you can, on a data event, process the data. So you don't have to wait for everything to finish. You can start doing something with that data right away. And the cool thing with streams is that then you can just use the pipe pattern. So you can actually pipe things together in the same way you would do on the Unix command line. So for example, here we are looking for a human genome, and we're piping to a JSON parser, and then we're just piping to the command line, the standard out. And so this makes it very easy to build pipelines to analyze genomic data and to combine all these pieces together. And streams are a first-class construct in Node.js. So how do we implement them? Well, the easiest way is use a module, for example, like Mississippi. Or you can subclass the appropriate stream class in Node.js, which is the hard way to do. And then you also get bind to the specific version of Node.js. So you should try to avoid that. You should use another module to write your own streams. So that way you're not bound to a specific version of Node. So for example, if you use the Mississippi module, this provides you functions to write readable streams, writable streams, transforms, DUPLEX, and to create pipelines. And I'm going to explain that in a second. You can also check this repository. Awesome Node.js Streams has a bunch of cool modules that are based around streams. It was created by our Google Summer of Code student. So with streams, you can process data in chunks. So you can have a readable stream. So let's say reading from a file, or a request, or just reading the standard in. And then you can pipe that to a transform stream, a parser, a filter. You can do some kind of multi-thread analysis, query to a database, anything. And then you can pipe that to a file or to standard out. So that's usually the types of streams you have. And what streams give you is that if somewhere in your pipeline you have a bottleneck, so you have, for example, network latency, or you have an issue accessing the database, you don't have to worry about timeouts or the pipeline crashing, because streams handle back pressure, which means that if one of the steps of the pipeline is slowing down, the whole pipeline, the whole streams adjust to that. So for example, if you have a big chunk of data that's blocking one of the steps, or you're waiting for some response of a database or request, the initial implementation of streams, you would lose data, because when they were push streams, so as you were getting data, you were not ready to read it, and so the data would get lost. So they changed to pull-based streams, which means that if your pipeline is blocked, when you try to do this push data, it returns false, so the data goes to a buffer. And if the buffer is full, the whole thing stops until downstream it gets processed. And then when that happens and downstream returns true, then you use a callback to get more data. And the cool thing is that you can do this push data several times. So you can split something into several chunks to send it downstream. And then when all the chunks are processed, you can use the callback. So what this would look like in code is, if you use a module like Mississippi, and Mississippi basically has true as a dependency, so instead you can just use true, the module true. The way to write a stream is you provide a transform function, and in this case we're using true object because we're gonna deal with JSON, but you could be dealing with buffers. And so the transform function is gonna get an object, an encoding. So what you do is you can modify that object. You can do whatever you want with it. So here we're adding a property to the object. And then once you're done, you push it downstream. And if you want to ask for more data, then you use the callback. So this is how you write a stream in 9 lines. Now, if you want to, for example, write a pipeline, let's say you wanted to filter some genomic data. So for example, here we are creating a filter function. So we're just going to look for the property sequence and see if it matches these 3 letters. It's kind of a dumb example, but it's just to illustrate how you could do that. And so if the object is a match, you push it. Otherwise, you just ignore it. And once you've done it, you've done everything you wanted, you call next to ask for more data. And so you can then use another module called Pumpify to combine all these streams in one. And so what Pumpify is doing is using the NCBI, the Bionode NCBI module to fetch from the nucleotide database, so which is called nuc core. And then it's piping whatever it gets to the filter stream that we just wrote above. And then you can stringify that and send it to standard out. So this is a very basic pipeline. And then you can write data to that pipeline. So here we're writing queries to Bionode NCBI, but that's basically how streams works, and you could be writing anything to it. Another kind of streams you can have are duplex streams. And what they are is when you combine a writable stream and a readable stream together. So it looks like a transform stream, but they're 2 independent streams combined. So it could be, for example, you're writing and reading from a database, or writing and doing requests. So it's 2 separate streams, but you can combine them together in one duplex stream. And Mississippi provides a method to do that. Another kind of streams that you can do are pass-through streams. And what they do is that they usually just look at the data. So for example, you could implement a counter with that, or you could do something. So they don't modify the data, but they do something else each time the data goes through. So for example, again, with Bionode NCBI and the true module, and let's say you have your own database module, you could implement a simple counter. And so you create an empty stream. So it's just, to implement a pass-through stream, you don't provide the transform function. And every time you have an on-data event, you can increment the counter. And that way you could pipe, we're doing a query to look for a genome, all the genomes are available on NCBI, all the spider genomes, and we're just counting them. And as all the data we get, we're writing to a database. So the counter doesn't touch the data. And once your script ends, you can console log the count if you want. And the cool thing with pass-through streams is that then you can use them to create forks in your pipeline. So you can reuse the same, observe the same data and do something else with it. And this is how you could, for example, implement some kind of multi-threading, or sending your request or your function, your whatever, to another machine or another cluster. And this is what it looks like. So it's the same thing. You just create a pass-through stream, and then here we are doing a query, and we're stringifying it, and then we're piping to fork. But then you can read fork for 2 different things. So one is piping to the command line standard out, and the other is writing it to a file. So that way you can create more complicated pipelines. So that was what I had to tell you about streams. If you're interested, check our project and contribute. And that's it. Thank you.